Yes, good morning students. In this video we are going to see the next practicals, that is analysis of drugs by chromatography method. Okay, now we'll move to the video. See, this is the heading, how to analyze the drugs. So okay, whatever drugs we are getting, like example cocaine, that is nicotine, so these are all considered as drugs, morphine, and then that is which are harmful drugs, okay, which are habit forming drugs. They can be analyzed by this chromatographic technique. Now see what we have to see. What are the materials needed? Materials that we need: goggles, okay, gloves, and then coming to the apparatus, they are centrifuge tubes for of 50 ml, chromatography paper, iodine crystals, pipettes, rulers in order to spot the points in the paper, okay, and then spot plates, that is the dimensions of 12 well, that is mentioned as dimension, okay, spot plate, and then gloves, we need gloves, then iodine fuming chamber, hair dryer in order to dry, okay, and then what are the specimens we need is the acetaminophenol, that is a tablet, that is acetaminophenol is nothing but paracetamol, and then aspirin, then specimen caffeine, okay, and then the specimen that is to be questioned, that is under our analysis, which is the drug which is under our analysis, that is the specimen. And then we need that is iodine crystals and UV light source in order to find out in order to detect the image, and then scissors and toothpicks are also needed. So these are the materials needed for our practicals. Now we'll move to the practicals, how to prepare the chromatography jars and strips. First point. So how to develop this? See, developing a chromatography strip may takes a few minutes to half an hour on temperature and other factors. To save time, we will prepare several chromatography jars in which we will develop all our chromatography strips at the same time. One strip per jar. So we'll use 50 ml self-standing centrifuge tubes as chromatography jars. We want these tubes to be saturated with the solvent vapor before we use them. So prepare them at least a few minutes before you intend to develop the chromatograms. Okay. Now first step is to remove the cap and remove the cap on one of the 50 ml. Okay. See, remove the cap on one of the 50 ml centrifuge tubes, stand the tube on a flat surface, and then use a pipette to fill the tube with 70% ethanol to just slightly above the top of the tapered portion. Now recap, now close the tube and set it aside and make it stand vertically. Okay, so this is the first step, and then repeat the first step to make additional chromatography jars. So you have to prepare a number of jars. See, if you don't have 70% ethanol, see next step, that is if you don't have 70% ethanol, okay, ethanol, then what you have to do is isopropanol can also be used, okay. Drugstores sell both these alcohols, that is they are called as rubbing alcohols. So that is ethanol, 70% ethanol, or that is in the place of 70% ethanol you can use isopropanol 70%, okay. But by the pure alcohol rather than a product that includes green dye or other contaminants. So that you have to check whether the product is a pure one. That one you have to check it, and you have to buy it analog samples. Instead of laboratory samples, LR samples, use analog samples so that the results may be accurate. Now, the next one. Now what we have to do is cut as many strips of chromatography paper to about 2.5 to 10 centimeter, that is 1 inches into 4 inches, as you need to run all of your specimens. So you have to cut the papers, okay, and then cut the papers. See the picture, you can understand how the paper strips are kept inside the beaker using a clamp, and so that we can spot the drugs, and so that we can analyze the drugs. Okay, see, after cutting the strips, the strips must be cut in a way that the width of each strip should be sufficient to allow the strip to stand freely upright in the tube without flopping into the content, plopping on contact with the sides of the tube. So it should not fall within it. Okay, so you must keep it in this way, or it is a better way to make it as a roll, okay, so that we can easily immerse it in ethanol. We can easily keep it in as ethanol. The length of the strip should be such that it comes as close as possible to the rim of the tubes without protruding above it. Okay. Next, use the ruler and pencil to draw two light lines across the width, that is of short dimension of each strip. One line about 0.5 centimeter from one end and the second about 1.5 centimeter from the same end. Place two hash marks on the 1.5 centimeter line, each about 1/3 of the width of the strip in from the edge. Okay, so next use the scissors to cut the corners of the strip from 0.5 centimeter line to the end of the strip. This will allow the bottom of the strip to protrude down into the tapered portion of the tube where it can be drawn up by the alcohol. Okay. Now, how to prepare the solutions of known and questioned specimens. Now our step after preparing the chromatographic jar, our step is to prepare the solutions of our known and questioned specimens. So we have the known compounds are acetaminophenol, and then that is aspirin and caffeine. These are all known drugs, and the unknown drugs we are keeping, that is the questioned specimens, they are also kept as a plot. Okay, chromatography is extremely sensitive, providing useful results with the specimens of 1 milligram or less. Accordingly, we need make up only a few drops of each solution. So it is very that is the analyzing part. It is taken only in a very small amount, that is a micro scale level. Okay, and now if you have not already pulverized acetaminophenol, what tablet you should not, that is if it is not, you must make it a small crystals, okay, small pieces, powder form, they have to pulverize it. Okay, transfer the tiny amount of acetamino powder, about as much as half of the gram of rice, to one of the spot plate. Add 5 drops of alcohol to the well. Record the contents of the well in your note. Okay, now repeat the first step to make the solutions of aspirin, acetaminophenol, along with that next one aspirin, and aspirin in well 2, and in aspirin that is aspirin that is caffeine in another well. Okay, that is in the spot plate we have number of wells. Okay, and that is the dip portion. In that dip portion we have to take each and every sample. Okay, one is acetaminophenol, and another is aspirin, and another is caffeine. Okay, if you have additional specimens, make up solutions of them also in the other wells. Okay, now we'll move to the next part. Next one is spotting and developing the chromatograms. How to spot it and how to develop it. Okay, now we'll see. The next step is to prepare the chromatography strips by transferring tiny amounts of the known and the questioned specimens. That is the process called the spotting. Once we have finished spotting the strips and allowing them to dry, we will develop the chromatograms in the chromatography jars we made up earlier. So this is the step we are going to follow. For these, what are the procedures we have to follow? See, what we have to do is, we have to first wear the gloves to prevent skin oil from contaminating the chromatography paper. That is very important. So we need gloves, because our fingerprints must not be there. Okay, so we must use gloves for all that is for revealing them latent fingerprints of other specimens. We must use gloves in order to avoid our fingerprints. Okay, place the first strip on a clean sheet of paper or other clean working surface. At the top end of the strip, use the pencil to label the strip, and then left side then is one side acetaminophenol, and then the next one will be next specimen will be aspirin, and the third one will be caffeine, and the last one will be the unknown sample, that is the question, that is the specimen that is to be analyzed. Now dip a clean toothpick into the solution, involve one of the spot plate and touch the tip gently at the left hash marks on the strip, transferring a tiny amount of the solution to the strip. Now our goal is to make small spots, only minute spots, ideally about 2 to 3 millimeter or less than that, centered on that hash mark, centered on them. Now I will show all these things visually also, okay, so that it will be more clear to you. After both spots have dried, repeat the steps, that is repeat that spotting one part, you allow them to dry, okay, and then you, after 2 or 3 minutes you spot the next one and allow it to dry. Like that you repeat it, okay. Repeat the steps again and again for 3 to 4 times, so that we can get more amount of that sample, okay. Then repeat you have to repeat the process until you have plotted uh, all your specimens, okay, that the known specimens and the unknown specimens. And then, okay, so this is the way we are plotting, okay, this is the way we are plotting, and that is the spotting and developing the chromatograms. Okay. Next, how to visualize the chromatograms, okay. How to visualize the chromatograms. Here at this point we have 3 or more developed chromatograms, each of which looks like a plain strip of paper with a few pencil marks on it. Only we can see like this. The analytes present on the strips are colorless and they are invisible. To visualize them we use ultraviolet. So for this purpose we use ultraviolet radiations or iodine fuming, or both the techniques can be used. Usually in the real forensic labs, or both the techniques are used, either using UV radiations or using iodine fuming. Okay, now so this is the way we will visualize the items. Now you can see these things in this. See what is happening here. See here. See what is this? You need whatever need, that is a chromatography paper and pencils and pen, and then a jar. Okay, jar containing, that is jar is with, first it is rinsed with the distilled water, and then it is and we are taking ethanol in it. Okay, and now what we are going to do is, see, these are the papers and the ruler. See how the value, how they are, how that is plotted. See here, it is even plots are kept, so that the distance must be even. So we are using a ruler. Okay, now this one in pencil we are marking it, and then later using this, that is now you see how the values are, how the drugs are plotted there. See here, now the mark is kept in order to find out the distance. If they are accurate, the values are kept accurate, they are kept at even distance. Yes, and now that is in order to differentiate. In order to differentiate what is kept, we are having color difference. Okay, now convert into a round, that is a cylindrical strip. The strip is converted into a cylinder, and so that elution can take place in an easy manner. Okay, here the eluent that is used is ethanol, and that will raise, that is taken along the plots. See, you can see the plots, and but here that is they had already shown is that is visualized one. Visualized chromatogram is shown here. Okay, but you have to visualize using ultraviolet radiations or iodine chamber. See the values, see this one, the value, see there's a known one, and this one is the unknown strip. And so from the, this is the solvent front, the marking is the solvent front, or we have to find out the distance from the edge to that of the solvent front, and then you have to see the RF values. RF value is the distance between the particular substance divided by the distance of the front, solvent front, and if you calculate that value, we can find out the substance that is present, and the drug can be analyzed from the known values. If the substance is kept or known, then we can have the known values. We can see the known values. um, and the last one is, using the iodine chambers and ultraviolet radiations, these chains can be seen very well. Okay, and the chromatograms can be seen, are very visible. Okay, thank you students.